In this video, I'm going to discuss about some questions based on molecular basis of inheritance. This is one of the most important chapters of biology, class 12. And students, I believe that if you want to recapitulate the lesson, so discussing question is the best way. So let's start. The first question is, in a DNA strand, the nucleotides are linked together by glycosidic bonds, phosphodiester bonds, peptide bonds, hydrogen bonds. So here the point you have to consider, that is the nucleotides, how nucleotides are linked together. So to solve this question, let's understand what is the structure of the nucleotide. This is the deoxyribose sugar attached to the nitrogenous base. Let's take adenine, and here we will number the carbon atoms. This is the first carbon atom, this is second, this is third, this is fourth, and this is the fifth carbon atom. Through the fifth carbon atom, phosphate group is attached. This is one nucleotide. Let's draw one more nucleotide. And these two nucleotides, they are joined together by diester bond. So how? So this, by removal of these two nucleotides, they will react and there will be removal of one molecule of water, and as a result of this, that is dinucleotide will form. So let's see the structure. During the reaction of the 2-hydroxyl group of phosphoric acid with a hydroxyl group in two other molecules, two ester bonds are formed. Two ester bonds you can see here, that is two ester bonds are formed. So these bonds are called phosphodiester bond. Other options were like the glycosidic bond. Glycosidic bond is present between the nitrogenous base and the sugar. And what is glycosidic bond? So glycosidic bond is a type of covalent bond that joins carbohydrate molecule to another group. This bond is the glycosidic bond. Then comes the peptide bond. Peptide bond is present between the amino acids, and the next hydrogen bonds don't link the nucleotides. So yes, it is present between the nitrogenous bases like adenine, and two hydrogen bonds are present between adenine and thymine, and three hydrogen bonds are present between the cytosine and the guanine. So our correct answer will be phosphodiester bond. So the second question says the size of the DNA of bacteriophage lambda is, and options are given. 50,000 base pair, 48,502 base pairs, 48,602 base pairs, and 58,000 base pairs. So means we have to calculate. So let's calculate. We have to calculate the lambda bacteriophage length in terms of base pair. So actually the length of lambda bacteriophage is 16.5 into 10 to power minus 6 meter. You know that the distance between two consecutive base pair is 0.34 into 10 to power minus 19 meter. So if we calculate the number of base pairs present in this much length, then we will get the answer. You have to divide 16.5 divided by 10 to power minus 65, 0.34 into 10 to power minus 9. You will get, if you do, let's see which one will be the closest, that will be the answer. So this option is the closest one. So right option will be 2. So here this is the correct answer. That third question says that is the nucleic acid is a polymer of option 1 nucleoside. It is formed by the condensation of sugar and nitrogenous base. Okay, So in the nucleic acid, you know that is there. It contains sugar, nitrogenous base, and the phosphate. So here that is the here only nucleoside, only two of the components are present. So this answer is not possible. So we will delete this answer. Then comes nitrogenous base. So not possible. This is only one component. So this is also not possible. Phosphate, only phosphate is not a component of nucleic acid. So that is this option is not possible. Then comes the nucleotides, and nucleotides contains all the components like nitrogenous bases are present, phosphate is present, sugar is present. So that is the right option is, that is D, and it is nucleotides, right? Now we will move to the fourth question. The component of nucleoside are, that is, let's first discuss that is what is nucleoside. Nucleoside means that is the sugar and what? Nitrogenous base. So these two components together form the nucleoside. Let's see the options. So option 1, pentose sugar and phosphate group. Not possible. Phosphate group is not here, so it's not possible. So this is the wrong answer. So so mark it as this is, delete this. Then comes the pentose sugar and nitrogenous base. This is the correct option. Next question, nucleotide contains. Chalo, nucleotide contains, what are the components are present in the nucleotides? So nitrogenous, I will add one more component here and we will get the nucleotide, components of nucleotide. So here I am adding plus phosphate group. So so here the first option says that is pentose sugar nitrogenous base phosphate group. Okay, so it seems correct. So before marking it as correct, let's see the other options. Pentose sugar nitrogenous bases. So no, here only two components are present, so this is not possible. Nitrogenous base and phosphate group, only two components, so this option is also not possible, and none, so this is also not possible. So our correct option is, that is first option, pentose sugar nitrogenous base and phosphate group. Sixth question is, adenine pairs with thymine with, here options are 1 hydrogen bond, 2 hydrogen bond, 3 hydrogen bond, 4 hydrogen bond. Between the adenine and the thymine, 2 hydrogen bonds are present, and cytosine and guanine, they are joined, linked together by 3 hydrogen bonds. So it will be 2 hydrogen, 1 hydrogen bond. It's not possible. So 2, it seems correct, but let's see the others. 3 hydrogen bond, not possible, and 4 hydrogen bond, this is also not possible. So correct option is 2 hydrogen bonds. Similarly, the seventh question says cytosine pairs with guanine. Options are 3 hydrogen bond, 2 hydrogen bond, 1 hydrogen bond, 4 hydrogen bond. So refer to this fact, and this is here cytosine and guanine. Cytosine and guanine are attached by 3 hydrogen bonds. So correct option is this. Nitrogenous base is present in DNA are, and we know that the adenine and guanine are purines, and purines, adenine and guanine are common to RNA and DNA, and pyrimidines like thymine, and cytosine. Thymine and cytosine are the pyrimidions present in the DNA, and of thymine, uracil is present in the RNA. So now see the options. The first says that adenine, guanine, cytosine, and uracil. So uracil is present only in case of RNA. So this option will be incorrect. Adenine, second option, adenine, guanine, cytosine, and thymine, all four, that is nitrogenous bases, which are present here in the DNA, are here in the second option. So this is the correct option. If we see the second, third, and the fourth option, C and D, that is the adenine, guanine, and cytosine. So only three different types of, that is the nitrogenous bases. So this is not possible. And here that is the uracil, and also three nitrogenous bases. So this is also incorrect. The question says which statement about polynucleotide chain is not true. A nucleoside has three components: nitrogenous base, pentose sugar, and phosphate group. We know that the nucleoside is formed by the condensation of nitrogenous base and the pentose sugar. So this option appears incorrect, but let's see. So for me, adenine and guanosine are purines. Adenine and guanosine. See, adenine is nitrogenous base and guanosine, that is the nucleoside. But it's a guanine is purine, so we consider this statement is correct here. And two nucleotides are joined together by phosphodiester linkage to form dinucleotides, which we have already referred in the first question. So this is also correct statement. A polynucleotide has a free phosphate moiety at had a free phosphate moiety. A polynucleotide has free phosphate moiety and hydroxyl at two opposite ends. So that is also correct option. If we draw the structure of DNA, it will be something like, it will appear like this. And this is the phosphate molecule here. So this will be phosphate molecule here. And then we can see that is the, again it is attached to the phosphate, and again here like this. And so this chain continues, and at the here, that is 3 at the third position, that it has free hydroxyl group. So the following is not true, or DNA double helix structure. Two nucleotides are anti-parallel polarity. So this is correct. So both runs in, that is the, in the both strand, they run in the opposite direction. They are parallel, but they run parallel and in the opposite direction. So this option is correct. Bases in two strands are linked by hydrogen bonds. Yes, that is the, here suppose that adenine is there, so that opposite will be, that is with two bond, hydrogen bond, it will be attached to the another nitrogen base like thymine on the other strand. So like that here, that is if it is cytosine, so with 3 hydrogen bond, it will be attached to the nitrogen base of another strand, that is guanine. So yes, this option is also correct. Two chains are coiled in a left-handed fashion. So this is wrong because it is right-handed. It is coiled in a right-handed fashion. The fourth option is, that is pitch of helix is 3.4 nanometer. So if we draw the helix, so here that one turn, that is complete turn, is called, that is the, represents the pitch. Incorrect option is this one, and remaining options are correct. I hope this video would have made you recapitulate your concept of DNA structure. In the next video, I will discuss some more questions based on the same topic. Please subscribe the channel and share and like the video. Thank you for watching.